Module 6, Cell Cycle, Part 1. I just wanted to share a story about this art of microscopy with this chapter opener picture. This brilliant colorful fluorescence micrograph shows a newt lung cell in mitosis. Centrosome stained magenta, microtubules in green, chromosomes in blue, and keratin intermediate filaments in red. The work was done in the lab of Dr. Alexey Khodyakov at Wadsworth Center, Department of Health, New York. The micrograph was taken in the year 2000, 20 years ago, with a modified Nikon TE200 regular fluorescence microscope. It is not an image from a confocal microscope, and you can appreciate the quality of the image. The image won the first prize in the annual Olympus Nature Light Microscopy Imaging Competition in the year 2000.
Now let's talk about the cell cycle in action. The cells reproduce themselves by duplicating their content and then dividing in two. The time it takes for this purpose to happen is known as the cell cycle. And the duration of the cell cycle differs in cell-specific manner, so it differs from cell to cell. For example, a fly embryo cell have a cell cycle of about 8 minutes, which is very fast. Whereas mammalian liver cells have a cell cycle that's greater than a year, so they don't divide very much. So this is a figure that shows a typical stages of the eukaryotic cell cycle, and these are the stages that you have seen before in other courses. Broadly, the cell cycle is divided in two phases, the mitotic or the M phase, which is relatively much shorter, and the interphase. The cells remain in the interphase much longer, which are divided in several phases. Before going into the actual phases of the interphase, I just wanted to point out that the cells may undergo into a G0 phase, shown here as an offshoot from the cell cycle after a mitotic division. It means a cell may decide to go into a quiescent stage and may not undergo active division, but upon a stimulus, it can again enter the cell cycle. Many cells in our body exhibit this behavior. Interestingly, your neurons are completely amitotic and they are permanently in the G0 phase and do not come out of it at all. So now let's take a look at the actual phases of the interphase. So in the G1 phase, also known as a gap phase, we can think of it as the first growth phase. And during the G1, the cell has come out of the mitosis and is growing, and it's monitoring its environment to make sure the environment is okay, that nutrient sources are okay, basically making sure all is okay. So the DNA can be replicated at this point after the G1, and the cell enters into the S phase or the synthesis phase. And during the S phase, the DNA is replicated. So this is the DNA synthesis phase. After the S phase, you have G2 phase, and G2 phase really serves as a kind of a safety gap, so the cell can make sure that the chromosomes have been duplicated properly or the DNA has been duplicated properly before the cell enters mitosis. Because you can imagine how detrimental it would be for the daughter cell if you did not have appropriate duplication of DNA prior to mitosis. And then finally mitosis or the M phase, this is when the cell divides. So visually, the most dramatic of all these phases in the cell cycle is the mitosis. Microscopically, you can see a lot of changes in the cell structure, in cell shape, and many people think that the rest of the cell cycle, which is interphase, is a kind of an uneventful interlude. But in fact, there are very complex processes going on during interphase to regulate the cell cycle. And I hope after this topic, you will appreciate how complex and how elegant those regulatory mechanisms are that are taking place during the interphase. So this is really an overview of how this topic is going to be covered. In the first part, we are going to talk about the mitosis, and really what we will be discussing in this part of this topic is what the microtubules are doing during mitosis. That will actually complete our conversation on the cytoskeleton, kind of a continuation of our microtubule conversation in the cellular cytoskeleton. Then we will talk about the regulatory mechanisms that are in place during interphase. And finally, of course, you can't talk about cell cycle without talking about stem cells and cloning, and we are going to cover those in parts 3 and 4 of this topic. So in this part, we are going to focus on mitosis, and mitosis itself can be divided into the 4 stages: the prophase, the metaphase, the anaphase, and the telophase. And I'm sure that you have heard of these before, but we will look into these phases in a little bit more greater detail. So in the first figure, we have the interphase, and during this phase, you get duplication of the DNA. Replication of the chromosome occurs during the S phase or synthesis phase of interphase. You can also get duplication of microtubules organizing center. Remember, the most microtubules in the cell emanate from a microtubule organizing center, and in the interphase cell, it usually looks like this. You have a single microtubule organizing center with all the microtubules emanating from that point. So you can get duplication of that microtubule organizing center. Then in prophase, you get the migration of these 2 microtubule organizing center known as spindle poles towards the pole of the cell. You can start to see the chromosome condensation, and you start to see the nuclear envelope disappear. In prometaphase, which is the very earliest phase of the metaphase, you get the capture of these chromosomes by the microtubules. And then as metaphase proceeds, you get an alignment of these chromosomes along the center of the cell. Next, the cell moves into anaphase, and it's during the anaphase where you see the 2 sister chromatids get pulled apart and they are distributed to either end of the cell. And during the telophase, you have the formation of this contractile ring, which is made up of actin and myosin, and you get a contraction event of the actin and myosin. Very similar to the contraction of the actin-myosin structure in muscle cells that we talked about, which leads to cytokinesis, which is the pinching off of the 2 daughter cells. So let's focus on the microtubules during mitosis. This is the organization of the microtubule during the metaphase, and together they form the mitotic apparatus. The mitotic apparatus has 2 main parts. There is this spindle-shaped part, which is in the football-shaped part in the center, and then there are microtubules that are arranged in tufts or bunches at either end of the spindle. They are called astral microtubules. And there are actually 3 classes or 3 types of microtubule in the structure. It is not that they are all different, they are all the same, they're all microtubules, but they have 3 different functions. So for example, the kinetochore microtubules shown here in the pink are the microtubules that are interacting with the chromosome. Then you have the polar microtubules shown here in the dark purple or in the blue. They don't interact with the chromosomes, but they are interacting with each other right in the center of the spindle. And finally, you have this astral microtubule, which form these tufts at either end of the mitotic apparatus. So it is very important for this chromosome to be attached to one kinetochore microtubule that's emanating from this spindle pole and one kinetochore microtubule emanating from this spindle. Because you can imagine how deleterious the consequence would be if, for example, the kinetochores of both of these sister chromatids were attached to the microtubules emanating from the same spindle pole. Because during the anaphase and during the cell division, one daughter cell would have an extra chromosome and one daughter cell will have one missing chromosome, and that could have serious consequences. These unfortunate events do happen, and which is known as chromosome aneuploidy. You all know that Down syndrome is a developmental disorder that happens due to the trisomy of chromosome 21. During the development of the gametes, one dividing cell receives one extra copy of chromosome 21, leading to this disorder. So the sister chromatids have to be oriented so that they separate, they move to the opposite poles, and this is known as bi-orientation. And the cell has a certain way to make sure that each sister chromatid pair attaches to the microtubules emanating from opposite spindle poles so that they are bi-oriented before metaphase proceeds into anaphase. Now let's take a look closely at how these microtubules are attached to this kinetochore region or the centromeric region of the chromosome. To be clear, the kinetochore region is the protein structure on the chromosome or on the sister chromatids where the spindle fibers or the microtubules attach during the cell division. So here is a detail of this chromosome. You have the centromeric DNA right here in the red, and it's associated with a very large protein complex known as the inner kinetochore protein complex. And that associated with another protein complex known as the outer kinetochore protein complex. And the plus end of the microtubules bind somewhere in this outer kinetochore region. There is also another region known as corona outside the outer kinetochore, which is not shown here. So please keep in mind that the inner kinetochore and the outer kinetochore, they are not individual proteins as depicted in this diagram, but they are actually large protein complexes. And here you can see an artificially colored transmission electron micrograph, and of those green microtubules interacting with the light brown outer kinetochore protein complex. It also shows the inner kinetochore with the centromeric DNA in the dark brown and the chromosomal DNA in the blue-black. Now we're going to look at these complexes in much more detail. This figure gives you a flavor of the complexity of the kinetochore region of the chromosome. You do not have to memorize the name of these individual proteins and functions except a few. The first thing that I want to point out is the organization of the microtubule to centromere attachment complex in a little more detail. You can see that the microtubules are attached to the outer kinetochore complex, traversing through the corona. Then you can see that inner kinetochore protein complex contain the centromeric DNA wrapped around the histone to form a nucleosome. We're going to talk about two proteins shown here in this figure. One is the CENP-A and another is this OR-B, which stands for the Aurora B kinase. So the take-home message from this figure is only the general organization of the structure and appreciation of the complexity. So how are chromosomes captured during the metaphase? I should say prometaphase to be exact. There are actually two ways in which they are captured. One is known as the end capture and the other is known as the side capture. So what happens during metaphase? You have these microtubules growing from the spindle poles and they are growing towards the chromosomes. And if one of these microtubules happen to, as it's growing, come in contact with the kinetochore region, it will bind to the chromosome and capture it. This is known as the end capture. But the cell is not always that lucky as this event happens by chance. And so the cell has another way to ensure that the chromosomes are captured. This is known as side capture. So what happens if you have a microtubule growing and it happens to miss the kinetochore region, so it doesn't bind to the kinetochore region? As it turns out that these kinetochore proteins can also interact with the side of the microtubules, and so it will capture the chromosome on the side of these microtubules known as the side capture. And then eventually these chromosomes will migrate or slide towards the end of the microtubule with the help of the microtubule motor proteins that we are going to see next. So let's take a look at how the chromosomes are captured first and aligned in the center of the cell, which is known as the chromosome congression, and the key players that are involved. So in the early metaphase or prometaphase, the microtubules elongate into the central spindle, as we have seen before, to set the stage of the event. So the first part is the chromosome capture, once again with a little bit more detail. So in the step 1 of the capture, the kinetochore can attach to the end as 1A, known as the end capture, or to the side of the kinetochore microtubules shown in 1B, known as side capture that we saw in the last slide. In the step 2, the chromosome is drawn towards the spindle pole by the dynein-dynactin motor complex that is associated with one of the kinetochore walking towards the minus end of the kinetochore microtubule. In the step 3, a kinetochore microtubule from the opposite pole, from the opposite pole attaches to the free kinetochore to biorient the chromosome. In the next step, the chromosome with only one kinetochore pole interact with the kinesin 7, also known as a SNP-E, and we'll see that what this interaction means in a second. So now these bioriented chromosomes with several kinetochore microtubules attached to this each kinetochore move to a central point between the spindle poles, and this is known as congression. So what happens after the congression of the chromosomes? There is an ongoing tug-of-war kind of thing going on, with one set of kinetochore microtubules shortening, whereas the others are elongating, which is known as the bidirectional oscillations of the chromosomes. On the shortening side, as you can see in the figure, kinesin 13 stimulates kinetochore microtubule disassembly at the plus end, a fact you can recap from our discussion on the microtubules in the previous module. Also remember that the dynactin-dynein motor complex moves the chromosome towards the spindle pole. You can also recap that the dynein-dynactin motor moves towards the minus end of the microtubule. On the other hand, on the lengthening side of the kinetochore microtubule, the kinesin 7 holds onto the growing microtubule. Note that the chromosome arms are still pulled or pointing away from the spindle poles owing to the associated kinesin 4, which is also known as the chromokinesin. The force of pulling to the poles overcomes the force of pulling the arms towards the center of the spindle, and that's the reason that they do have these V-shaped appearances. So I mentioned that the chromosomes have to be appropriately oriented. They have to be bioriented in order for the sister chromatids to be distributed to daughter cells appropriately. And the cell regulates this through 3 protein complexes. So I mentioned earlier that the kinetochore region of the DNA is marked by a specific histone, and this histone is known as SNP-A. So remember that DNA is not naked in the cell. You should remember this from your biochemistry. It is organized by its interaction with the histones, and one of the histone subunit is known as the histone 3, and one particular variant of the histone 3 is this SNP-A, and that particular variant of histone 3 is only found at the kinetochore region of the chromosome. So SNP is a kind of marks the kinetochore region for the mitotic apparatus. So here you have your DNA that is bound to the kinetochore region that has specific variant of the H3 histone, the SNP-A. Then you can see that it is associated with the inner kinetochore protein complex, and here is the outer kinetochore protein complex, and here is your microtubule in the green that is interacting with this outer kinetochore protein complex. So SNP-A is an important player because it marks the kinetochore. Another important complex is this NDC-AT complex. NDC stands for nuclear division cycle protein complex. The NDC-AT complex interact with the microtubule on one end and the inner kinetochore region on the other end in the sleeve-like fashion, and it forms like a tension. So this is a very rigid structure. So you have this sleeve made up of this NDC-AT complex that connects the microtubule to the chromosome. The third major player is a protein complex known as the CPC, which stands for chromosome passenger complex. And within this complex is a kinase known as the Aurora B kinase. And remember, kinases are the enzyme that phosphorylate other proteins. So what happens is that the CPC associates with the inner kinetochore early in mitosis, and the Aurora B kinase, shown here in the red in the CPC complex, is sitting right here at the kinetochore region and is able to phosphorylate many of the proteins in the inner kinetochore and the outer kinetochore. It can phosphorylate the NDC-AT complex, and when that happens, the association of the microtubule and this region is quite weak, or there is no or little tension. So in a more detail, here in the bottom figure, the aurora B kinase is in the red and the NDC80 is in the blue, and it is phosphorylated. But when this biorientation occurs, each of the kinetochore region on each sister chromatid is bound to the opposing microtubule, and the tension forms, and you get a change in the conformation of the chromosome, such that the kinetochore protein complexes are now pulled away from the CPC, or the chromosome passenger complex. And when that happens, the aurora B kinase cannot phosphorylate the proteins within this kinetochore region. That causes the association of the NDC80 complex with the kinetochore proteins to become very, very strong. It stabilizes the chromosome and it creates a tension. It is very stable structure. And that is really how the cell makes sure that the chromosomes are bioriented in metaphase, because if they have other kind of orientation, bound only to one microtubule or bound to microtubules from the same side of the mitotic apparatus, you are going to have the phosphorylated proteins in the kinetochore. And you are going to have a very weak interaction, and you are going to have a very weak association with the microtubules. Whereas if it is properly oriented, you are going to have a very strong association within this microtubule due to the fact the phosphorylation events are no longer being maintained by the aurora B kinase. Okay, so let's look at the behavior of these microtubules during the metaphase and anaphase. During a metaphase, the kinetochore microtubules, in other words, those that are interacting with the chromosomes, maintain a constant length. So these chromosomes are aligned in the middle of the cell. So you might imagine that this is a static structure. But in fact, it is not. The treadmilling is going on. So you are getting constant addition at the plus end, and it is the plus end that is interacting with the kinetochore region. So you are getting constant addition at the plus end, and it is the plus end that is interacting with the kinetochore region. And you are getting constant subtraction at the minus end. And we have seen this before when we discussed microtubules. But the rate of subunits on is equal to the subunit off, and that is why you get the maintenance of this constant length of these kinetochore microtubules during metaphase. And if you could track, if you could mark these two subunits, for example, by a fluorescent dye, you could see that the subunits appear to move towards the spindle pole because treadmilling is going on. But during the anaphase, the microtubules, all three populations of microtubules, behave very differently. And the way they behave creates the forces necessary to pull the sister chromatids apart and to elongate this spindle apparatus. And these forces can be grouped or classified into two groups known as Anaphase A and Anaphase B. And the forces that are generated in Anaphase A are generated by the kinetochore microtubules that are interacting with the chromosomes. And the forces that are generated during anaphase B are generated by the polar microtubules that are interacting with each other in the spindle and the astral microtubules on the either end of the spindle. Now, I need to make a point that they are called anaphase A and anaphase B, but they are not temporal nomenclature. So in other words, it is not anaphase A happening first, then anaphase B. They are all happening at the same time. It is just easier to talk about them separately. So first, let's have an overview of the events, and then we will go into the detail. So in anaphase A, you get a rapid shrinking of the microtubules. You get loss at the plus end. Actually, kinesin 13 interacts with the plus end of the microtubule that destabilizes them, and you get a rapid depolymerization with the plus end. That causes those microtubules to shrink or appear to shrink towards the spindle pole. And when that happens, it pulls the sister chromatids towards the spindle pole. In the case of anaphase B, you have the polar microtubules interacting with each other where they overlap. They actually push against each other and push the poles apart, which causes an elongation of the spindle. And then you have the astral microtubules, which are attached to the plasma membrane of the cell and will actually pull on these spindle poles, which will further enhance the elongation of this structure. Now, if we look at more closely how these polar microtubules and these astral microtubules are contributing to this elongation event during anaphase, you will see that it's all carried out by the action of microtubule motor proteins, the kinesins and the dyneins. So here in this overlap region of the polar microtubules, you have some kinesin. The tails are interacting very tightly with one polar microtubule. The head of that kinesin is walking towards the plus end of the microtubule on the neighboring polar microtubule. And that causes these two microtubules to push against each other, and that causes the elongation event. On the other hand, these astral microtubules interact with these dyneins. The tail of the dyneins are bound tightly to the plasma membrane. The heads of the dynein in the green are walking along these astral microtubule towards the minus end, and that causes a tension to be set up and pulls the astral microtubules such that you get even farther elongation. And there is even evidence that there is some polymerization of the microtubule that are going on in this overlap zone, which further elongates the whole spindle apparatus. Now here is another detailed rendition of the events in Anaphase A and B and the role of the motor proteins. So in Anaphase A, the movement of the chromosome to respective poles are powered by the microtubule shortening by kinesin 13 proteins at the kinetochore, and this is happening at the plus end of the kinetochore microtubule shown as A1 in the figure. In addition, kinesin 13 also aid in disassembly at the minus end of the spindle. The kinesin 13 just make the disassembly faster in addition to the normal disassembly of the microtubule at the minus end, which is shown in the A2 of the figure. Also note that, as I have mentioned earlier, that the chromosome arms still point away from the spindle poles owing to the associated of kinesin 4. Due to the force pulling the poles overcome the force pulling the arms towards the center of the spindle. In Anaphase B, the sliding of the anti-parallel polar microtubules is powered by kinesin 5, which is a plus end directed motor as shown in B1. Finally, the astral microtubules grow towards the periphery and stretch the cells. In contrast, the dynein-dynactin motor attached to the plasma membrane in the cell cortex try to walk towards the minus end of the spindle pole and generate the pulling force to stretch the astral microtubules and elongate the cell for division. And so this is really all I wanted to point out about mitosis and the role of microtubules in this process.
So this concludes Module 6, Part 1.